Hello and Assalamualaikum. My name is Alif Irfan bin Muhammad NAWI, and I'm going to present to you about my FYP2 project, which is isolation of endophytic fungi from AZADIRACTA indica and the screening of their antimicrobial activity.
Okay, let's take a look at the background. Azadirachta indica, also known as name plant, is an indigenous medicinal plant. Despite their activities, several parts of the tree, including bark, fruits, and leaves, have a medicinal and industrial application. Endophytes are a renewable supply of bioactive substances that can be used in medicinal, agricultural, and industrial. Endophytic bioactive compounds such as antibacterial and antiviral have properties that have been recently identified. Although antibacterial and fungal can be extracted directly from the plant, this study would like to investigate the ability of the endophytic fungi living within the plant as an alternative source of antimicrobial products. First objective is to isolate the endophytic fungi obtained from the indica extract. The second objective is to study the antimicrobial activity of endophytic fungi isolated from extract of A indica. Next is we will talk about the Azadirachta indica. So there's example of the classification of the A indica, and then for the geographic location, it's an indigenous plant that can be found in India and Africa. Other than that, it can be found in Pakistan, Sri Lanka, and also in Asia. As for the habitat, it can invade shrublands, woodlands, or grassland. For the bioactivity, some examples are anti-inflammatory, antioxidant. And then for the morphology, it has distinct features which is like flowers, leaf, fruits, bark, and branches.
Okay, next is endophytic fungi. The word endophyte refers to all microorganisms, specifically pathogenic bacteria and fungi. They do not cause any disease or other negative effects. And then fungal endophytes can survive in plant tissues and can be beneficial to the plant itself. For example, the biological activity that can be found is antioxidant, anti-inflammatory, antibacterial, and antifungal. For the species of endophyte fungi that can be found within A indica is, for example, Phomopsis species, Aspergillus and Penicillium pavum.
To achieve the study's objective, we began with the sample preparation. Neem leaves were collected from three leaves, young, matured, and foliate. These leaves were then sterilized by separating and surface sterilization. After that, we will proceed to the isolation of endophytic fungi. The purpose of isolation of endophytic fungi is to obtain a pure culture of isolated fungal endophytes. And then the fresh pure culture were obtained after incubate. This step ensured we isolated the endophytic fungi from the leaves. And then once we had a pure fungal cultures, we will perform the antimicrobial screening. In the primary screening, which is the Agapta assay, was used to identify fungal isolates with potential antimicrobial activity. The most promising isolates was then tested in the secondary screening using a distribution assay with a crude fungal extract that we have done by fermentation of solvent-solvent extraction. The inhibition zones were measured to assess their effectiveness against various test microorganisms. Finally, we will conduct a statistical analysis using a one-way ANOVA to compare the inhibition zones and determine whether the observed antimicrobial activities were statistically significant or not. Next is results discussion. Um, from the experiment, there are a total of 4 fungal endophytes were successfully isolated from leaves. According to the results, matured leaves yield the highest number of isolates, highlighting that the suitability for the endophytes collection. This is likely due to their longer exposure to fungal colonizer compared to young or foreign leaves. Thus, for optimal isolation of endophytic fungi from mature leaves are typically the best choice. Okay, the next part is the primary screening agaplaceae. In the primary screening, isolate IMN1. It display a significant activity against gram-positive bacteria, including subclass P. cereus and S. aureus, and also yeast and alkans. So based on the results, isolate IML1 was chosen as the most prominent endophytic fungi to proceed with the distribution assay. A one-way ANOVA test analysis was conducted and the results obtained were significantly with P less than 0.05. And then after the couple assay, the design discussion is about the secondary screening, which is distribution assay. At 100 milligram per ml concentration of crude endophyte extract sample, almost all test bacteria exceeded the degree of susceptibility towards isolate IML1. The most susceptible for gram-positive is S. aureus. Meanwhile, the least susceptible is P. subtilis. And then for the gram-negative bacteria, E. coli is the most susceptible, while S. typhi is the least susceptible. For yeast, only Candida albicans is susceptible, and for Cryptococcus neoformis, there were no results of it. Okay, next is the continuation of the discussion about this diffusion acid. Studies show that gram-positive bacteria are more susceptible to antibiotics than gram-negative bacteria. This is likely because gram-negative bacteria have an extra outer epithelial layer, providing additional defense against antibacterial agents. Chloramphenicol demonstrated strong broad-spectrum antibacterial properties, effective against both gram-positive and gram-negative bacteria. On the other hand, ethyl acetate showed no activity, aligning with studies indicating its role as a solvent without antibacterial properties. Interestingly, the crude extract from the fungal endophyte was effective against Candida albicans, whereas the positive control, the fluconazole, was resistant. This supports findings that endophytes may exhibit stronger antifungal effects due to their unique mechanism. Finally, a statistical analysis using a one test confirmed a significant result with p-value is less than 0.05, highlighting the potential of endophyte as a powerful source of. Okay, so here's the last part of the results discussion, which is the statistical analysis by using one-way ANOVA from question Excel. As you can see, there are two analyses that have been done, which is the crude endophytes extract sample and positive control. By using one-way ANOVA test analysis, both have been conducted and then the results obtained were significant. P-value is less than 0.05. In conclusion, a total of 4 endophytes have been successfully isolated, with isolate I and NL1 being the most prominent endophytes. Isolate I, M, and L1 was determined when almost all test bacteria and yeast were susceptible, and the results were deemed significant. He suggested that it provided an important information that could further influence the production of naturally derived antibacterial and antifungal obtained from a INDICA only through its fungal endophytes. For the recommendation, first you can conduct a minimum inhibitor concentration MIC, minimal bactericidal concentration MBC, and minimum fungicidal concentration FC analysis to further determine the effects of the lowest concentration of antimicrobial agent that can inhibit or kill microorganisms. And then second is conduct a fungal staining for simple identification of the isolated endophytic fungi. Lastly, conduct a phytochemical evaluation for the identification of secondary metabolites secreted by fungal endophytes that possess the antimicrobial properties.